This investigation is titled Searching for Bioactive Molecules in Prostate Cancer from Mayan Traditional Medicinal Plant. I'm Juan Trinidad. I work as a researcher in two Uruguayan labs, Laboratory of Bioactive Molecules and Laboratory of Molecular Interactions. Now, I'm going to tell you about the development, result and perspective of these investigations. Before that, we want to say thanks to the organizer and the scientific committee for selecting us to diffuse and present our work. It's a great honor for us to be participating in the first International Electronic Conference on Medicinal Chemistry. In this slide, I leave you the abstract of the research. Much of this will be contained in the talk, so let's move on. Cancer is a generic term for a large group of diseases characterized by the growth of abnormal cells beyond their usual boundaries that can then invade adjoining part of the body and or spread to other organs. Cancer can affect almost any part of the body and has many anatomic and molecular subtypes that each require specific management strategies. Cancer is the second leading cause of death globally and accounts for 8.8 million deaths in 2015. Lung, prostate, colorectal, stomach, and liver cancer are the most common type of cancer in men, while breast, colorectal, lung, cervix, and stomach cancer are the most common among women. Here we focus on prostate cancer. Prostate cancer is the second most frequently diagnosed cancer in men worldwide. This type of cancer is the fifth leading cause of cancer death, with the highest mortality rates found in the Caribbean and Southern and Middle Africa. In more developed countries, prostate cancer is the most frequently diagnosed cancer and also one of the leading causes of cancer death among males. The basis of prostate cancer is not well understood, but the androgen receptor is known as a key in development, evolution, and survival. In the last decade, the treatment used for excellence is androgen deprivation, which, in turn, has side effects on the hope and quality of life of the people treated. Also exist other treatments as surgery, radiation therapy, hormone therapy, and chemotherapy. Usually, some of them are combined depending on the progression of the disease, but all of them have side effects. At this moment, several researchers have shown that plants are source of new metabolites of potent pharmacological activity in prostate cancer, such as the case of capsaicin, curcumin, Taxol, and others. This way of investigation has really good results. An example of that is Taxol derivatives, particularly docetaxel and cabitaxel, that are currently used in chemotherapy as the first line of action in different kinds of cancer, including prostate cancer. We propose this approach using plants from Mayan traditional medicine, an ancient indigenous culture of Central America. Folk medicine is used around the world in different cultures, such as African, Indian, and South American. It's based in natural products and hundreds of years of experience. Actually, there are contemporary indigenous communities like Mayan that treat sick people with some success using this medicine. This community has more than 3,000 years of experience and was one of the biggest and more developed indigenous societies of America. Their knowledge has not been deeply studied yet. Our group of investigators pretend to use medicinal knowledge as a source of new drug development. To find active molecules in prostate cancer, we start interviewing the traditional healers called shamans. From them, we have obtained a variety of 30 plants with medicinal properties, but actually we are performing our assays with 10 plants. These plants were selected from more than 300 species that they use as a treatment for a variety of diseases, including cancer. First, methanolic and dichloromethanolic extraction was done. The active extracts were subfractionation firstly by silica gel chromatography and secondly by preparating thin layer chromatography. To select the active plants, we test the extract and subfraction in a cancer androgen-dependent cell line called ecmc. To test all extract, we test all extract with a fixed dose of 25 micrograms milliliter. The cells were exposed 24 hours to the compound and cell viability was measured by MTT assay. The MTT assay is a colorimetric assay for assessing cell metabolic activity. Not pH-dependent cellular oxidoreductase enzyme may reflect the number of viable cells present. MTT assay involves the conversion of a water-soluble MTT to an insoluble formazan. The formazan is then solubilized and the concentration determined by optical density at 570 nanometers. This is what we call bio-guided fractionation. The process was done sequentially. NMR and MS were used to elucidate the active molecules. Based on this new molecular structure, we designed synthetic derivatives to enhance antineoplastic activity. This graph shows cell viability of ecmc cell line exposed to methanolic or dichloromethanolic extract of different plant organs grouped per plant. Gray column shows ecmc cell viability of control DMCO used as reference. Out of 22 extracts, 14 have relevant activity. It should be noted that the active plants were in all the extracts with some exceptions. Black arrows shows selected samples to continue with the bio-guided fractionation. Here we show the 4 plants that we use more extensively and the reported properties. Nidosculus chayamansa is widely used in Mayan folk medicine in a variety of diseases. Scientists have reported low acute oral toxicity, anti-inflammatory activity in mice, as well as antioxidant activity in vitro. Out of 4 selected plants, only capsicum has already tested in prostate cancer cell lines, showing interesting anti-proliferative activity. Because of that, we select to validate methodology. The other 2 plant extracts were tested in normal and different neoplastic cell lines, showing selective toxicity against the latest. It's worth to know that they were not assayed in prostate cancer. Down in the slide, you can see the isolated organic compound from each one. One of the selected plants was Leucaena leucocephala. Up here, the graph shows cells viability, our control, and all the fractions get from silica gel chromatography assayed in ecmc cell line. We get 3 leader fractions, but from here we continue with F28 and F29 that were subject to a preparative thin layer chromatography. This is the results of the thin layer chromatography of F28 and 29. The next plant is called Chacha by Mayans, but their scientific name is Nidosculus chayamansa. Above in the slide, the graph of the subfractionation by silica gel chromatography of T3 that correspond to a methanolic extraction of Nidosculus chayamansa's leaves. From here, we continue with F58 and F31. The first one were subject to a preparative thin layer chromatography. The results of the latest is shown down here in second graph. As you can see, The best fraction of preparative cell layer is F9, with approximately 50% of inhibition. Then we perform the structural elucidation. The structure is shown here with pictures cell line apoptotic phenotypes. This is compound C isolates from F9. Later, we test our recently isolated molecule, named compound C, on 4 different cell lines in a fixed dose of 133 micrograms. The first one, PPH1, corresponds to a prostate epithelial cells from a benign prostate hyperplasia. The other 3 are cancer cell lines ordered based in the aggressiveness. This graph shows silica gel chromatography of Terminalia catapa. In this case, the inhibitory activity present in the complete extraction was being lost, so we don't continue with fractionation. The fourth selected plant was Capsicum Chinese. Here, the graph shows the subfractionation by silica gel chromatography of T31 that correspond to a methanolic extraction of Capsicum Chinese fruits. From here, we continue with F0 and F11. The last one was subject to a preparative thin layer chromatography and then to NMR. The result of the last test is shown here in green graph. F0 was directly subject to NMR. From F11, we get capsaicin, a molecule with reported activity in cancer. From F0, we obtain a novel molecule similar with the obtained from Cnidosculus chayamansa. The isolated structures is shown down here. After obtaining all the previous structures and with the objective of improve the inhibitory activity of the selected molecules, we designed some synthetic derivatives based on said structures. In the image, we show 7 capsaicin derivatives and their activity on our cell line model LNCAP. Most of these have no relevant activity, but the compound D improved the activity of its precursor capsaicin by approximately 4-fold. This compound inhibits 89% viability of LNCAP in a 48-hour assay with a concentration of 58 micromolar, as you can see on graph. This is the molecular structure of compound D. In left side, a steroid group, and in the right side, the pharmacophore group of capsaicin. So maybe you are asking yourself, why a steroid group? Well, LNCAP is a cell line that corresponds to a cancer phase and androgen dependent. So we use this group as a carrier to androgen receptor. Also, this lipophilic group will rise diffusion through cell's membrane. In conclusion, we test 10 plant species from Mayan folk medicine and found that 9 of them have cytotoxicity activity in prostate cancer cells. We perform a bio-guided fractionation to isolate the active compounds, validating this process by the isolation of capsaicin from Capsicum Chinese fruit. We also describe 2 more compounds, one from Capsicum Chinese fruits, compound B, and another from Capsicum chayamansa, Cnidosculus chayamansa, compound C. Also, we designed 7 new compounds inspired by nature. One of them has an AC50 lower than 25 micromolar, compound D. Then we identify a new hit for the drug development process.